Hi, I'm Sam Durrance, and I'm going to be talking about our NanoRacks project, which we call Self-Assembly in Biology and the Origin of Life. But to start out with, I thought I'd say a little bit about our school. We feel like we're kind of one of the local universities. In fact, Florida Tech was founded in 1958 by some of the engineers that were here in Florida to help establish the space program here for NASA. It's now a university with colleges in engineering, aeronautics, business, psychology, arts, and sciences. And the project that I'm going to be talking about today really involves two of those, the Engineering College and the College of Science. Let's see. Currently we have over 13,000 students, on extended campuses and the main campus. We have about 7,000 or so students on campus from all 50 states and from 100 countries. It's a fairly international university. Roughly 25 to 30% of our student body are international students. We now have a football team, so that makes us legitimate, I think, as a university. And in fact, last week we won. So let me talk a little bit about the project itself. This is a project that, as I said, is called Self-Assembly in Biology and the Origin of Life. A bit of a reach for what it is we're trying to do. But what we're studying are self-assembly processes that self-assemble under thermodynamic control, rather than all life processes, where assembly takes place primarily under the control of enzymes, very things we call it kinetic control, things like that. This project was started by a few people in the College of Engineering and College of Science, myself from Science, Hector Gutierrez and Daniel Kirk from the College of Engineering. We had a lot of student involvement in this project to begin with. There were undergraduate students from 3 different societies on the university, the Student Rocket Society, the Society for Physics Students, and the Students for the Exploration and Development of Space. At one point we had about 15 undergraduate students working on this, helping us develop the proposal. It's a student-driven project. We are currently have 3 graduate students working on the project. We have 2 of them here with us. We have Jose Avendano. He's in the College of Engineering, working on the design of the experiment, and Dylan Bell in the College of Science, working on the scientific aspects of the experiment. So let me say a little bit about what it is we're trying to do. And I should also point out that this is an experiment that originated in this building about a decade ago or so, in one of the laboratories down on the first floor of this building. So it's sort of maybe coming full circle here. We were growing amyloid fibers, and I'll say a little bit about those. You see the 3 pictures here. We have we can grow in vitro in a test tube. These fibers, these are lysozyme fibers. Lysozyme is a hen egg protein, and it will, if the conditions are right in solution, it will form these very long, thin fibers, which are as narrow as one protein, a couple of nanometers in diameter, and can be as long as 15 or 20 microns long, and they form spontaneously if the conditions are right. We want to study this process in microgravity, and in order to study it, there are a number of variables that can alter the growth of the fibers, and so what we decided to look at was just the time variation. They grow through a series of stages, and in the laboratory that's quite easy. We just form the solution and the buffer solution. We put the protein powder in it, and then we sample it every day or so, or every few days. Now we can't do that in orbit, so we wanted to build an automated laboratory that would fit in one of the NanoLab cubes. And so what we decided to do was we have 9 small vials, and the vials, in order to initiate the aggregation of the proteins, we have a buffer solution, and we have to place the protein powder in the buffer solution and raise the temperature to 50 degrees C. When that happens, then that initiates the growth phase, and they will grow until we turn the temperature off, essentially like that. And so originally we were planning to have 9 vials, and we would start one at the beginning of our on-orbit time and just allow it to run to the end, and then we would periodically start them. It turned out that we didn't have quite enough power to do that, so we've come up with another strategy where we still cover the same time series, but by running either 3 or 4 vials at the same time all the time. So we have in the experiment itself, we have 9 vials that keep the buffer solution separated from the protein powder until we get ready to start initiation of aggregation. And I'll describe that a little bit more in an upcoming slide. We, of course, are going to be flying this to the space station. This project, by the way, has been moved to SpaceX 5. We ran into some problems in the development of the vials themselves. And let's see. Here's a blow-up of the experiment itself. And it's composed, as I said, of 9 vials. These are the vials shown here. And on each vial, there's a linear stepper motor that drives a piston into the fluid to start the protein aggregation itself. And we have 2 PC boards that control the vials themselves. There's a nichrome wire heater that's wrapped around the outside of the coil. And there's a heater controller on the PC boards. And there are thermocouples that measure the temperature of the fluid itself so that we can bring it up to 55 degrees C. And let me show you just some of the components here. This is the heart of it, the vial itself. And it's a fairly complex little piece of engineering. And it's one of the things that sort of held us up in the development itself. Not so much the mechanical design of the vial, but the fact that these protein fibers are very sensitive to the material under which they're growing. We have to control the pH accurately and raise the temperature and hold it at 55 degrees C. And it turned out that originally we were going to make them out of polypropylene with some metal. There's a floating piston here, which is sealed with O-rings. The fluid goes into this part of the chamber here itself. There is a piston up here that is driven into this fluid, and in order to take up the pressure that would have been the increase in pressure if we just drove that piston down, this piston floats, and so it moves away to maintain the same volume. We also the solution itself is acidic, and so there are safety concerns. We have to make sure that it has at least 2 levels of containment. And so this is a cross section of the piston itself. And one of the problems that we found is we have to make everything out of polypropylene, all of the pistons and everything, because the proteins just wouldn't grow in anything else other than all polypropylene. Even to the point where at one point we had the thermocouple coming in from the bottom piston and moving into the fluid with some epoxy around it. And that stopped the growth of the proteins themselves. So we had to come up with a different design where it's completely enclosed in polypropylene, and the thermocouple is inserted in there with some thermal paste around it so that we get good thermal contact. Each of the moving parts has 2 O-rings, or these are actually X-rings because they have to slide. And so that's, in principle, 2 levels of containment, although we found out during the integration process that may only classify as 1 because we can only test it as both O-rings together and not individually test them. But we also have the level of containment of the nanoracks itself. That will be sealed, and there's a little air relief valve on it so that the pressure will equalize inside. Let's see. So this part around the outside is a little confusing. That's actually not part of the vial. That's just the background. This is the edge of the vial itself. The nichrome wire is round around here. This is the floating piston with 2 O-rings. There's a sleeve here that's used so that we can install the protein powder in this little slot right here. We install that first and assemble that completely. Then we put the proper amount of buffer solution in this part of the chamber, We push the floating piston up, and then we start inserting this sleeve with the piston itself downward. This responds and goes back until it's in this sealed position, and then it's ready to be installed between the upper and lower plate and install the stepper motor on it. So that's the way this piston works. And that, as I said, that was quite a challenge. We've run through about 4 different complete designs, and we have manufactured them all by now. There are 2 PC boards. There's the PC board on the front we call the vertical PC board. It contains a microprocessor for controlling everything. We also use an internal clock because we want to be sensitive to any power outages on the system. So we have an internal clock that runs a cadence, essentially, to tell which valves to turn on when and to turn off. And so that's controlled by an internal clock. This board also has the connectors for the thermocouples around the outside, and the controllers for the thermocouples, and then there are also the connectors for the nichrome wire that wraps around each of the valves themselves. And lastly, we have what we call the horizontal board, which has the connectors and controllers for all of the stepper motors on it itself. Say a little bit now about what it is we're hoping to learn. As I said, if we look at this, this is a kind of a time series of lysozyme fibers grown in the laboratory. They go through a series of stages. Initially, we began to form small units of either areas where we have not 2 fibers, but multiple fibers wrapping around each other, forming a large system itself. So all in all, this to us is a very exciting proposition. We wish we were flying tomorrow night, but we're not. We're going to fly on SpaceX 5. We're still in the process of finishing the development of the payload itself, and so that's it. Thank you for this opportunity as well.